Hello everyone, welcome to another video by IGCSE Passion. For this video, I'll be talking about Chapter 9, which is Transportation in Animals. So if you're new to this channel, don't forget to subscribe, and without further ado, let's start. So according to 2023 syllabus and onward, it will be covering these four sub-chapters of Chapter 9. I'll be covering it all in this video. If you're still having some questions, don't forget to write it in the comments below. Let's start. So, circulatory system. The definition of the circulatory system is a system of blood vessels with a pump and valves to ensure one-way flow of blood. So, we have the blood vessels, which are more like a tube-like structures that carries the blood to a pump, which is the heart, which is made up of muscles that will press the blood and move it to another direction or to another flow according to the side that we're talking about. And the valves are more like a door structure that controls the flow of the blood. So about circulation, we have two types of circulation. We have the single circulation and the double circulation. So you need to know the difference between single and double circulation. You need to know why is double more efficient from a single circulation, and where can you find each of the circulations. So starting with a single circulation, it is found in fish mainly. You cannot find it in a frog, for instance, because frogs, when they grow up, they would live on land rather than water, so they cannot really uh, use single circulation while they are on land. Now, fish have a one blood clotting chamber, which is the atrium, and one blood ejection chamber, which is the ventricle. Pressure is lost as the blood passes through the capillaries of the gills and is not built back up again until the blood returns to the heart. This makes a single circulation inefficient. So, explaining it again, um, now the blood passes through the heart once as oxygenated, or let's say there's nothing like the human heart, so it would enter the heart as let's say deoxygenated. It would reach the gills and then it goes to the body, but there's no pressure that the blood would face. So the pressure that have been given to the blood inside the heart is lost when it passes through the body, and so less pressure will be going through the fish body, and so it is inefficient. But talking about double circulation, which is found in all animals and humans, uh, the right side of the heart collects the deoxygenated blood from the body and pumps it to the lungs. The left side collects the oxygenated blood from the lungs and pumps it back to the body. The double circulatory system helps to maintain blood pressure and making circulation efficient. Now, talking about the heart structure and what do we need to know that the vena cava gets the blood from the body into the heart, and it would get it right into the right atrium. Now, after the right atrium, it would get stuck in its place by the atrioventricular valves, where it closes, and then it would open. After it opens, the blood would fall into the right ventricle, and from the right ventricle, it would be pumped into this tube structure, which is the vessel, which is the pulmonary artery. And what does it and how is it controlled? It is controlled by the semilunar valve, so the blood won't go back into the heart. Now, after it reaches the lungs and goes back, it would go through the pulmonary vein here, and then it would enter into our left atrium. Now, after entering the left atrium, it will be stuck in place by atrioventricular valves. After that, it would fall into the left ventricle, and after being after it is stored in the left ventricle, it will be pumped into the aorta to reach the body. Now, there are some structures that are not really related to the blood flow. It is mainly related to the heart functioning. So, the septum, which separates the right from the left side. So, how can you imagine the thing? Like, how can you make sure that you're not making any mistake? Imagine that the heart is like it is flipped by a mirror. So, when you look at the mirror, your left hand and your right hand are flipped. The same goes to your heart. So the left for you is the right for the heart, and the vice versa for the right side. So we have the base, which is uh, this red part, the very red dark part, is known as the muscular wall, which will cause the pressure. Moving on, the wall of the left ventricle is much thicker than the wall of the right ventricle. Why is that? Because it needs to build up enough pressure to pump the blood to all the main organs. But the right ventricle pumps blood only to the lungs, which is a shorter distance. So according to the distance, the pressure would differ, and of course, the thickness of the muscle wall would differ as well. So the wall of the atria are much thinner than those of the ventricles. Why is that? This is because the contraction of the atria only needs to be powerful enough to move the blood down into the ventricles, while the ventricles are moving blood around the body and through all the organs. Okay? So talking about the septum that I showed you earlier, which is this structure that separates the right from the left side. So it divides the left side of the heart from the right side. Why is that? To prevent mixing of deoxygenated and oxygenated blood. Moving on, blood is pumped away from the heart in arteries and returns to the heart in veins. So a way to recall these names is by using this way. I've been using it during my O levels. So arteries start with an A, and the way starts also with an A. So whenever they say an artery, they mean that the blood is flowing away from the heart. While for the veins, there is two letters, which is the I and the N, which are coming beside each other, making the word N. So it is going into the heart. Cool, right? Moving on, there we need to know the activity of the heart may be monitored by different ways, starting with the ECG that you can find in many hospitals, like these um, lines that you can see, the structures. These are known as the ECG. Um, Now, there is a question that I found earlier in past papers which contained the structure, and they're asking about how many times the heart pumped, I guess. I'll be solving this question later on. I'll be solving classified questions based on the latest syllabus. I'll be making classified papers from 2020 to 2023. So stay tuned so you can watch it later on. Now, going to the other way to monitor your heart rate is by using a pulse rate. Now, the difference between an ECG and a pulse rate is that the pulse rate won't show you the sequence of the heart pumping, or like how the how the heart is pumping with a certain pace. But for the pulse rate, they are showing you how many times it would pump and how uh, like the amount of pressure that is going inside your heart. Now, lastly, we have the valves closing sound. So you can detect the sound of the valves on your breast and in a certain area in your neck. You can notice this sound or this vibration that happens, I guess, from the valves, so you can know exactly your heart rate. But the most efficient ways are the pulse rate and the ECG. Moving on, we're going to talk about the physical activity. So, a heartbeat is a contraction. Wait, let me use a highlighter. So, it is a contraction, each of which forces blood to lungs and body. The heart rate increases from resting rate and stay high until physical activity slows down. or stops after exercise, the heart rate gradually returns to normal. So it won't go immediately to normal state, it would require time, and I'll be talking about it later on, I guess in chapter 11 or 12. I'm not really sure, but it is most probably chapter 11, which is talking about the gas exchange in the body. So it is more related to the gas exchange. Now, going deeper through the physical activity, during exercise, the heart rate increases. Why is that? To supply the muscles with more oxygen and glucose. These are needed to allow the muscles to respire aerobically. This is very important. It's not anaerobic, it is aerobic. Aerobic requires oxygen, anaerobic does not require, so they have sufficient energy to contract. So muscles need energy, how they can receive it by aerobically respiring and receiving oxygen and glucose. Now, talking about coronary heart diseases. Coronary, sorry, I'm saying it wrong. So, what is a coronary heart disease? It is the blockage of a coronary artery. Now, can you see these blood um, vessels that are going outside the structure of the heart? These are known as the coronary heart uh, arteries. So um, they are blood vessels that are outside the heart. They supply the heart with the required glucose and blood. So when they get blocked, a human can die from that. So risk factors contains diet, sex, genetics, age, lack of exercise, and stress. So diet, if you're not having a balanced diet, eventually your heart won't be healthy. Depending on your gender, so from a gender to another, you may be having more risk to have a coronary heart disease. Genetics, so it is related to your family genes. Age is age, stress, and lack of exercise. How to prevent having coronary heart disease is by maintaining a healthy, balanced diet with a lower chance, uh, will lower the chance of a person becoming obese. So, being less obese means that the low intake of saturated fats that is part of a balanced diet reduces the chance of a build up fatty deposits and the formation of blood clots. So how does that happen? I guess there's a photo right here. So see these yellow structures that are blocking the way of the artery? It is like a fat, it's a fat layer. So when it builds up, it will prevent the red blood cells to pass through it, and that can cause a blood to get blocked, to block this artery, and so uh, there would be a heart shock that would happen. Now going to the blood vessels, um, so we have three types. We have the arteries, veins, and capillaries. Arteries carries the blood at a high pressure away from the heart. Wait, away from the heart. We said how we can know if it's away or into the heart to the organ of the body. Then we have the veins. Veins returns the blood at a lower pressure from the organs to the heart. And lastly, capillaries. Capillaries link arteries to veins. They carry blood through organs and tissues, allowing material to be exchanged. Now, arteries carry blood with a high pressure, as we said earlier. It carries oxygenated blood, have a thick muscular wall to 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 bypass or to overcome the pressure that is happening inside it, and it have a very narrow lumen to have a high pressure, and lastly speed of flow is very fast. But the veins carry blood at a low pressure towards the heart, carry deoxygenated blood other than pulmonary vein, have a thin wall, have a large lumen, contain valves, but arteries don't, and lastly, veins have a slow speed of flow. Lastly, capillaries. Capillaries carry blood at a low pressure within tissues, carry with oxygenated and deoxygenated, have walls that are 1 cell thick, have leaky walls so that the material inside it can't pass and reach the tissues easily. And lastly, speed of flow is slow. Wait, I cannot highlight it. Okay. Going on, the liver is served by hepatic artery. You need to memorize that. It also receives blood through the hepatic portal vein, bringing uh, products of digestion from intestine. Blood leaves the liver via the hepatic vein. Heart limited to vena cava. Uh, you can go back in this video and check the photo that I explained earlier. You can find all of these that I mentioned them, but I didn't explain them. But the heart contains vena cava, aorta, pulmonary artery, in the pulmonary vein. Just for your knowledge, vena cava is a vein, aorta is an artery, lung limited to pulmonary artery and pulmonary vein. Pulmonary is related to the lungs, and the coronary is related to a heart. So whenever they say coronary, just know that it is talking about something about the heart. When they mention pulmonary, they are talking about the lungs. Kidneys is limited to renal artery and the renal vein. Now going to the blood, which is our last part. Blood is made up of a liquid, which is the plasma, containing red blood cells, RBD. Wait, let me just write in the short form. I'm telling you, do not write it in the short form. Write it fully because they may not give you the mark. So red blood cells and white blood cells and platelets. Wait, let me use our highlighters for that. Give details of the components of the blood, their functions. The term used for naming the white blood cells are the lymphocytes, and the phagocytes are needed only for extended paper, and I'm talking about the extended paper. So starting with the red blood cells, it is a biconcave disc shape with no nucleus, which carries the oxygen. Now going to our two white blood cells, which is the lymphocyte and the phagocyte. So a lymphocyte is having this weird large structure inside there, which is the large nucleus. It produces antibodies. Why? To fight bacteria and foreign material, while the phagocyte would engulf. So you can see this lobed nucleus so that it can engulf, like it can just curve the shape and just get the material that it wants to engulf into it. So fight diseases by surrounding the bacteria and engulfing. The process is called phagocytosis. Lastly, platelets. Platelets is related to the blood clotting process. Okay? Now, the blood plasma is a liquid that transports substances to cell and carries waste away. Oxygen combines with hemoglobin to form oxyhemoglobin. The oxygen is released from the red blood cells in capillaries where surrounding oxygen levels are lower. So mainly the red blood cells carries the oxygen. It is a biconcave shape. There's no organelles in there. Why is that? To have more space for more oxygen. And what also? It contains hemoglobin to combine it with the oxygen. Thirdly and lastly, which is the platelets, we need to know the blood clotting process. Platelets clump together with tissues are when tissues are damaged and block the smaller capillaries. The platelets and damaged cells at the wound produces a substance that acts on soluble plasma protein called fibrinogen, and the result it would turn into an insoluble fibrin, which forms a network of fibers across a wound. Red blood cells become trapped in the network and so form a clot. The clot is not only stopping further loss of blood, but also preventing the entry of pathogens. So we need to know about the clotting process is that it would turn from soluble to insoluble state, so fibrinogen would turn into fibrin and it would create a mesh or a network of fibers. And the process of clotting is to reduce the blood loss and to prevent pathogens from entering into your body. And that is all for today. Hope I helped you and have a nice day. And don't forget to subscribe.